I hope you're all doing well and you're healthy. Thank you from the students that told me the number of power. Very thank you, Mr. Qasim Mahavi Haydar Qasim Mahavi. Thank you for your attention. Uh, you helped me very much. Thank you. Microbial phylogeny, we are going to continue from this power, and today we are going to start the power number 3, but your lesson and your sections are not behind. I told you before, and I don't want to teach more than 3 power or 4 power, I think, and that is why I teach a little less each section. Okay, microbial phylogeny is the title for today. The phylogenetic tree representing all living organisms shows that evolution of current forms of life occurred from a common ancestor, the universal ancestor. Okay, so the phylogenetic tree representing all living organisms. You know, all living organisms are included in phylogenetic tree, and the phylogenetic tree is originated from a common ancestor cell, a common cell that we call that ancestor cell, a universal cell. They are just the same, a common ancestor cell or a universal ancestor cell. Okay, so here you can see the picture. Here you can see the picture for the phylogenetic tree. We have 3 domains here: bacterial, archaeal, eukaryote. Okay, so we have 3 domains here, and we see a universal ancestor here. A common cell, universal cell, is the origin of all 3 domains of life. We have 3 domains. The bacteria and archaea domains are prokaryote, and the eukaryote is eukaryotic domain. So we have just 1 eukaryotic domain and 2 prokaryotic domain, and all of them are originated from a universal ancestor cell. Universal ancestor cell, and eukaryote domain is concluded with plants and animals. Okay. Two domains are of prokaryotic systems of life, the archaea and eubacteria, in contrast to previous systems of classification, wherein the prokaryotes were confined to a single kingdom. 2 domains are prokaryotic systems of life. Two prokaryotic domains, the archaea and eubacteria. You know what's the meaning of eubacteria? Eubacteria means real bacteria, not archaea. Every bacteria is except archaea are eubacteria. So eubacteria is all of the bacteria, not included archaea. In contrast to previous systems of classification wherein the prokaryotes were confined to a single kingdom. However, it's intriguing to note that genomic studies have shown the archaea to contain unique gene sequences which are not present in bacteria or eukaryotes. Certain genes are also shared between all the three domains. So the archaea have contain unique gene sequences that are not present in bacteria or eukaryotes. The genes are specific to archaea. They have specific genes for themselves in the archaea domain, and the genes are very different with bacteria and eubacteria. Certain genes are also shared between all the three domains, and in spite of this explanation, some genes are also shared between three domains. So we have common genes too. Okay. The divergence of the organisms respects the differences. The divergence of the organisms respects the differences in genetic sequences which could have become fixed in each group as they evolved. It is also postulated that earlier HGT played a key role in transfer of genes between organisms early in the evolutionary history. So that was a question in final exam, I think a term ago, that the question was which factor is playing a key role in transfer of genes between organisms, and the answer was HGT. You know, HGT is brief for horizontal gene transfer. So the answer was horizontal gene transfer. And the HGT is used for transfer of genes between organisms, and maybe the question was, I don't know, I don't remember accurately, but I know that HGT was a question. Maybe the question was which factor is important in evolutionary of microorganisms. HGT, please notice, mutation, and HGT play a key role in transfer of genes between organisms. Mutation and HGT are two factors, are two elements that, two factors better, mutation and HGT or horizontal gene transfer are important in evolution of microorganisms. It occurs as a responds to any change in the environment and provides for better adaptation. So we can see many genes can be transferred by HGT or mutation, and these genes that are derived from another bacteria, another organisms, can be useful for responses to any changes in environments and can be useful for better adaptation of them. And then the next title is Prokaryotic Microbe, Bacteria, and Archaea. If you have any question, you can ask me from these problems. If you don't have any question, I can continue. Okay, so we continue the next title, the prokaryotic microbes, bacteria, and archaea. The archaea are considered to be the most primitive and are common inhabitants of the so-called extreme habitats, hot springs, deep sea hydrothermal vents, alkaline and acidic habitats. So here we are talking about just prokaryotic domain. The prokaryotic domain are contained bacteria and archaea. They are most primitive and common, but the archaea we know are the most primitive organisms in the world, and we call them extremophile too, because they can live in extreme habitats such as hot springs, with hot temperatures, with high temperatures over than 70 or 80 degrees centigrade, deep sea hydrothermal vents, alkaline and acidic habitats. They are all the habitats that they can grow in. And though the bacteria and archaea share certain common features, the archaea also share similarities with eukaryotes. So the bacteria and archaea have common genes and common features, common traits, but they differ from another traits. So they have similarities and differences and similarities. The species concept is another title we are going to study. The species concept. Currently, a polyphasic approach is used to define a microbial species using phenotypic and genotypic properties. Whenever a new taxon is proposed, it is essential that the organism be isolated in pure culture and its characteristic features. be tested under standard conditions. So, as you read before, a polyphasic approach or a polyphasic taxonomy is used to define a microbial species. You studied before, and we had read about polyphasic taxonomy. Do you remember? Uh, phenotypic polyphasic contains two properties: phenotypic taxonomy and genotypic taxonomy, that we have read it before. And a new taxon is very important and essential to be found, because new genes and new proteins are exist in them. And whenever we find a new taxon, we must first of all prepare a pure culture from that, and then we investigate their characteristics and traits. Okay. All the strains within a species must show similar phenotypes. That was the last sentence here. If the 16 sRDNA sequences of organisms are less than 98.7% or 97% identical, they are members of different species. So, if the 16 sRDNA sequences of organisms are less than 98.7%, we cannot call them in similar species. They are in different species. Or less than 78%, they are not in same species. And the last title in this power is Polyphasic Taxonomy. When classifying a new taxon, it is essential to describe phenotypic, genotypic, and phylogenetic information as accurately as possible. This continues the polyphasic approach of taxonomy and is shown in figure 8. Okay, we read about polyphasic taxonomy before. Here we are going to tell about polyphasic taxonomy completely. So, as we told, this kind of taxonomy is polyphasic because it is a method that investigates, that study the genetic and properties and traits all together. So we investigate phenotypic, genotypic, and phylogenetic information all together, and then we have a completely taxonomy. Okay, you can see at this picture we are going to describe this in another powers. Please notice in this picture, I'm waiting for your questions. If you have any question, you can ask. Okay, dear students, mm, I will give you a 5-minute break. I have a emergency call, so I give you 5 minutes break and then I come back to the class. Okay, so for 5 minutes later I will teach you. Thank you. Okay, dear students, hi again. We are going to explain about polyphasic taxonomy right here, and you can see this kind of taxonomy here in this picture. We have two kinds of taxonomy here. You can see phenotypic methods and genotypic methods here. So we are investigating two kinds of properties in an organism: phenotypic characteristics and genotypic characteristics. We call this method polyphasic taxonomy. Phenotypic methods are contained. These methods that you can see in this picture, and genotypic methods are contained of these methods here. So phenotypic methods are concluded classical. Please note, we are going to study each method separately in the next power. Okay, the power number 3 that I will teach you, each method is explained in that. Okay, so please note here, phenotypic method is contain classical properties such as colony characteristics, biochemical, physiological studies. So in phenotypic methods in polyphasic taxonomy that is mixed for phenotypic and genotypic together, the phenotypic methods concludes classical methods. Classical characterization such as colony characteristics, such as biochemical, physiological studies. And the next method for phenotypic investigating for phenotypic characteristics studies is numerical taxonomy. Number 3, cell wall composition. Number 4, fatty acid analysis. Number 5, isoprenary prenomes. Number 6, whole cell protein analysis. Number 7, polyamines. Number 6, cytochromes. Number 9, advanced spectroscopy methods, FTIR, pyrolysis mass spectrometry, UV resonance Raman spectrometry. All these 9 methods are phenotypic methods. Okay. Yes, this table, this chart is very important because each method is described in the next power separately. We have to read each method separately. For example, the most of techniques, not all of them, but the most of techniques such as cell wall composition, such as fatty acid analysis, we are going to study them. So this is very important, yes. Okay, and the genotypic method um, included 16S rDNA, DNA-based content, DNA hybridization, RFLP, RAFT, AFP, ARDRA, neurotyping, red PCR, and MLSD. These techniques are very, very important, and they are techniques that we call them genotypic methods in polyphasic taxonomy that we are going to study in the next hours. Okay, the phenotypic information comes from the colony characteristics, cell type, cell wall type, pigmentation patterns, proteins, and other chemotaxonomic markers, while genotypic features are derived from the nucleic acids, DNA, RNA. Phylogenetic information is obtained from studying sequence similarities of the 16S RNA or 23S RNA genes in case of bacteria and 18S RNA in case of fungi. And that is very important. We can study phenotypic, genotypic, and phylogenetic information all together in this method. And this method, this table is very important. Yes, Mr. Ali Hamid. This table is very important because we are going to study each method separately in the next section. So phylogenetic information is obtained from 23 sRNA genes in bacteria and 18 sRNA in case of fungi. That was the final exam question 2, please note. And here we are going to explain this. If you have any question from this table or from these things, you can say and ask me. Okay, I was just waiting for you. If you don't have any question, I can continue because that table was very important, and I told you please note at this table. If you don't have any question, I can continue. Polyphasic taxonomy and about the methods that we are going to study. Okay, phylogenetic techniques. The phenotypic methods are all those that do not include the DNA RNA sequencing or their typing method study of morphological characteristics, and chemotaxonomic profiles is broadly associated with phenotypic characterization as I told you. So here you can see uh, about phenotypic techniques, and about every characteristics that I told you before. Okay. Classical cloning characteristics by chemical and physiological analysis was the first method that we are going to describe as the uh, last one, last slide in this power. Classical cloning characteristics by chemical and physiological analysis. Okay, the morphological, biochemical, and physiological characteristics provide in-depth information on a taxon. The morphology can include the colony characteristics, color, shape, pigmentation, production of slime, etc. Further, the features of the cell are described as to shape, size, gram reaction, extracellular material like capsule, presence of endospores, flagellar presence and location, motility, and inclusion bodies. Light microscopy is generally used to describe the broad cell features. Okay, so that was the explanation of colony characteristics by chemical and physiological analysis that we are going to study. Okay, if you don't have any questions, I'm going to upload the new cover, and in a few minutes you don't have my voice because I'm changing the cover. Okay, so I'm going to upload another PowerPoint number 3. Okay, dear students, hi again. I was uh, changing the color. I changed the color of the power for you for better observe, because that was not black and white, that was colored, and it took a minute for changing the color. Numerical taxonomy. Number one was classical characteristics, phenotypic, and characteristics about colony, about mortality, about some characteristics in figures, endospores or mobility that we read in the previous power. Power number 3 is here the second method. If you remember, in the phenotypic method was numerical taxonomy. In polyphasic taxonomy, in that picture, the second method in phenotypic method was numerical taxonomy. Analysis of huge volumes of phenotypic data to derive meaningful relationships amongst a large number of microorganisms can be carried out using computer programs. This system of analysis is called numerical taxonomy. You're welcome. Yes, I know the color in black and white is better, and I was changing the color. Okay, the analysis of phenotypic data is very important in this technique, numerical taxonomy, and you know this technique is done by computer programs. This system of analysis is called numerical taxonomy. Giving numerical weightage to each trait is followed by analysis of the data by the computer programs, generating data matrix between each pair of isolates according to the degree of similarity. So as you see in this picture, in this power, we have some programs in the computer that it numer for us these characteristics. For example, trait number 1. For example, producing a spore is number 1. Mobility, mobile is number 2. Non-mobile is number 3. For example, smooth colony, the cover colony, number 4 or number 5. Each trait is numer, and then the similar traits in different bacteria automatically with program in computer can be numered. Okay, this is called numerical taxonomy. So we can see any data between each pair of isolates and the similarity between species. Based on the similarity, data cluster analyses are carried out. Based on different algorithms and dendrogram trees are generated showing the overall pattern of similarity, dissimilarity among the various organisms being studied. So based on the similarity data, we can draw, we can carry out the algorithms and dendrograms. You know, dendrograms are phylogenetic trees that we can draw them by some programs in the computer, and then we can study the various organisms together just at the same moment. While 16 sRNA sequences have garnered attention in recent times, the sole means of bringing out the uniqueness of a species, numerical taxonomy based on phenotypic traits of a large number of species compares favorably with that of phenotypic data. So please note that the 16S rDNA sequences are very important for drawing the phylogenetic tree. Okay, we must see the similarities between near species, and we call these similarities phylogenetic relationship. So when we talk about phylogenetic relationship, that means the studies concluded in phylogenetic tree. So numerical taxonomy based on phenotypic trait of a large number of species compares favorably with that of genotypic data. So here, with numerical taxonomy, we can study the phenotypic traits, and then we can compare these phenotypic traits with genotypic data. Okay, so don't forget this method. This numerical taxonomy is a method in phenotypic method. We can just investigate, we can just study, we can just find new traits, and we can just find and study the similar traits, phenotypic traits, not genotypic traits. Okay. And the third method was cell wall composition. Okay, I'm not teaching you more today until the power number 3 is enough, because your lesson is not behind, and I don't want to teach you very much, because the final exam will be hard for you, and so the final exam will not be easy for you. So I'm go slowly each section, and I don't want to gather the very explanation and texts for you. So I think that's it for today. Until cell wall composition. I have taught you. Please, Mr. Qasim Mahawi, please write the number of slides today too, and please write power number 3, slide number 3, power 3, slide 3. Okay, we are on the power 3 and the slide 3. Thank you very much. I hope for you a nice week, and I think for you healthy. Thank you, Mr. Ali Hamid, and all of you students. Take care of yourself, and goodbye until the next week. Bye bye.